everyone, in this video we will discuss about the flow cytometry. So first, what is the flow cytometry? Flow means flowing, cyto means cell, and metry means measure. To measure the flow of cell, that is the flow cytometry. Okay, so it is a popular cell biology technique that utilizes laser-based technology to count, sort, and profile the cells in heterogeneous fluid mixture. Using flow cytometry machine, cells or other particles suspended in a liquid stream are passed through a laser light beam in a single file fashion, and interaction with the light is measured by electronic detection apparatus. As light scatters, fluorescence intensity. Okay, so if a fluorescent label or fluorochrome is specifically and stereochemically bound to a cellular component, the fluorescent intensity will ideally represent the amount of the particular cell component. So this is the overview of the flow cytometry. Okay. Next is what is the exact mechanism or how the flow cytometry works. Okay. So number of measurable parameters that can be used by this technology to separate the cell population is immense, starting from simple surface immunophenotype to metabolic functions, then cell cycle status, redox state, and DNA content, the name few. Since its inception, FACS has been used extensively in biomedical research and clinical diagnostics. The flow cytometry instrument consists of three core systems, which include fluidic system, then optic system, and electronic system. First one is the fluidic system. Includes a flow cell where the sample fluid is injected. The flow cell required sheet fluid to carry and align the cells or particles so that they pass through a narrow channel into the laser beam in a single file. This hydrodynamic focusing allows the analysis of one cell at a time by laser intervention. The purpose of the fluidic system is to transport particles in a fluid stream to the laser beam. To accomplish this, the sample is injected in the stream of a sheet fluid, usually a buffered saline solution, within the flow chamber. And the design of the flow chamber allows the sample core to be focused in the center of the sheet fluids, where the laser beam then interact with the particle. Focusing is achieved by injecting the sample suspension into the center of sheet liquid stream. The flow of the sheet fluid moves the particle and restricts them to the center of the sample. So next one is this is all about the fluidic system. Next one is the optic system. Okay, so here optic system is located, which consists of various filters. Okay, then the light detectors and light source. Okay, light detector and light source, which is usually a laser line producing a single wavelength of light at a specific frequency. The optical system of the cytometer consists of excitation optics and connection optics. The excitation optics consist of laser and lenses that are used to shape and focus the laser beam to the flow of the sample. Next one is the collection optics. Consists of a collection lens to collect the light emitted after the particle interact with the laser beam and a system of the optical mirrors that divert the specified wavelengths of a collected light to the designated optical detector. A After a cell particle passes through the laser light, the rays emitted on the side of the fluorescent signals are directed to the photomultiplier tubes, and this is also called as a PMTS. And photodiode collects the signals. Lasers are available at a different wavelength ranging from the UV to far range, have a various range of power level as well. To achieve the specificity of a detector for a particular fluorescent dye, a filter is placed in front of the tubes which allows only narrow range of wavelength to reach the detector. Next, last system is the electronic system. Okay, the electronic system converts the signal from detector into the digital signal can be read by the computer. Here, once the light signal strikes one side of the PMT or the photodiode, they are converted into relative number of electrons that are multiplied to create a more significant electric current. The electric current moves to the amplifier and is converted to a voltage pulse. The highest point of the pulse is achieved when the particles strike the center of the beam, in which case maximum amount of light scatter or fluorescence is achieved. ADC, that is the analog digital converter, converts the pulse to the digital number and the graph is generated. So there are the different cell parameters which is used to analyze in flow cytometer, which include FSC system, that is the cell size system, then the SSC system, that is the side scatter system, and the last one is the fluorescent labeling of cell surface or intracellular structure using the fluorescent antibodies. So here FSC system is given, okay, which show the small angle scattering in the range of 0.5 to 5 and usually used for the size and shape. Next one is the SSC system. SSC system shows the large angle scattering in the range of 15 to 150 degree. Okay, which is mostly used dark field complexity and granularity determination. So the difference between FSC and SSC is given here. Forward scatter system is show diffracted light. Side scatter system shows the reflected light. For FSC system is used to determine the size of the cell. SSC system is used related to the cell granularity and determine the cell complexity. Okay. Then it is related to cell surface area FSC and SSC related to cell granularity. Okay. Next is the results of the FSC. So there are the two different flow cytometry analysis systems are used. First one is the single parameter analysis, and second one is the two parameter analysis. Single parameter analysis shows the histogram plot. Horizontal axis is used for the level of the fluorescence brightness, that is the brighter cells further light. Vertical axis is for the number of events per channel number. Analysis level of expression of the marker. Okay. Next one is the two parameter analysis. We show the dot plot graph. One axis shows the first color, second axis shows the second color. It is used for the analysis of individual population of cells. The next session we learn about the what is the exact method for the sample preparation, advantages and types of the flow cytometry.